尊敬的麦可卡琳院士，尊敬的谭广亨副校长，尊敬的各位来宾、各位老师、同学们，大家下午好。
Dear Professor Michael KARIN, dear Vice President Paul Tan Guangheng, dear distinguished guests, teachers and students, good afternoon.
欢迎各位来到校庆学术讲座。
Welcome everyone to the anniversary distinguished lecture.
我是今天的司仪，来自大学艺术团的练家英。
I'm today's host, Xu Xingyi, from the MUSD Art Troupe.
首先，请允许我介绍今天到场的主要嘉宾，他们是
First of all, we would like to introduce today's guests. They are
澳门科技大学副校监、校长李行伟讲座教授
Chair Professor Joseph Hong Wei Li, Vice Chancellor and President of Macau University of Science and Technology.
澳门科技大学副校长谭广亨讲座教授
Chair Professor Paul Tan Guangheng, Vice President of Macau University of Science and Technology.
澳门科技大学校董会校董邓君明先生
Mr. Deng Junming, Member of the University Council of Macau University of Science and Technology.
澳门科技大学咨询委员会委员曾智慧博士
Dr. Zeng Zhihui, Member of the University Advisory Committee of Macau University of Science and Technology.
澳门科技大学澳门发展经济学研究所所长雷鼎明讲座教授
Chair Professor Lei Dingming, Director of Institute of Development Economics.
以及现场的嘉宾朋友们，欢迎你们的到来。
And all our distinguished guests, let's welcome them again with a warm applause.
今天我们非常荣幸地邀请到美国国家科学院院士、美国国家医学院院士、澳门科技大学荣誉医学博士麦可卡琳院士，为我们带来主题为“炎症在人体健康与疾病中的角色：善恶与丑陋”的校庆学术讲座。
Today, we are very honored to invite our distinguished speaker, Professor Michael KARIN, member of the U.S. National Academy of Sciences and the National Academy of Medicine, Honorary Doctor of Medicine of Macau University of Science and Technology, will give us a lecture on the theme of inflammation in human health and disease, the good, the bad, and the ugly.
现在，让我们拍摄一张全场大合照，有请摄影师移步至台上。
Now, let's take a group photo with the entire audiences.
请大家望向镜头，合影留念。
Please look at the camera and take a group photo.
3、2、1。谢谢大家。
Thank you.
在讲座开始之前，让我们首先有请澳门科技大学谭广亨副校长为本次讲座致辞。
Before the lecture begins, let us first invite the Vice President of Macau University of Science and Technology, Chair Professor Paul Tan Guangheng, to deliver an introductory speech for this lecture.
Thank you. Ladies and gentlemen, distinguished guests, and esteemed colleagues, it is an absolute privilege for me to introduce our speaker for the 24th MUST Anniversary Distinguished Lecturer, Professor Michael KARIN, our triple academician. Professor Michael KARIN earned his Bachelor of Science degree in biology from Tel Aviv University in 1975, and subsequently a PhD in molecular biology from the University of California, Los Angeles in 1979. He has been the Distinguished Professor of Pharmacology and Pathology at the University of California, San Diego School of Medicine since 1986. Professor KARIN is an elected member of the U.S. National Academy of Sciences, the National Academy of Medicine, the American Association for Cancer Research Academy, and the European Molecular Biology Organization. This underscores the immense respect and recognition he has garnered in the scientific community. Professor KARIN's research has made a significant impact in our understanding of the intricate relationship between inflammation, cancer, and metabolic diseases, all very important subjects. His work has unveiled critical signal transduction pathways, illuminating the molecular mechanisms underlying these debilitating diseases. His research has contributed to cancer prevention, therapy, and the treatment of non-alcoholic steatohepatitis. Professor KARIN's transformative impact on the field is evident through his authorship of over 300 papers, with more than 100 of these in elite journals such as Nature, Cell, and Science. The significance of his research is underscored by over 300,000 citations and an H index of 275, a figure that most of us could only dream of. Today, we're indeed fortunate to have Professor KARIN share his knowledge and insights with us. Please join me in extending a very warm welcome to Professor KARIN as he takes the stage to deliver this distinguished lecture titled Inflammation in Human Health and Disease, the Good, the Bad, and the Ugly. Thank you.
感谢谭广亨校长的精彩致辞，请谭副校长留步。
Thanks to Vice President Paul Tan for his wonderful speech. Please stay.
接下来将进行一个简单而隆重的纪念品致送仪式。
A simple but grand souvenir presentation ceremony will be held below. May we invite Professor Michael KARIN to come on the stage? May we invite Vice President Paul Tan Guangheng to present a souvenir to Professor Michael KARIN and take a group photo.
有请谭副校长向麦可卡琳院士致送纪念品，并合影留念。
Please look at the camera. 3, 2, 1. Thank you.
谢谢。
Vice President Paul Tan, please be seated. Professor Michael KARIN, please stay.
请谭副校长移步至台下就座，麦可卡琳院士请留步。讲座现在正式开始，有请麦可卡琳院士。
The lecture is officially starting now. Let us invite Professor Michael KARIN, please.
Ladies and gentlemen, distinguished guests, dear Vice President Lee, I really thank you for making me a member of your community and inviting me, welcoming us, and asking me to give this lecture on a topic that is very close to my heart. But before I start, I actually have a question for the audience. How many people think inflammation is bad? How many people think inflammation is good? Okay, so I'll try to convince you that inflammation has many facets, many faces, and I'll start with some basic introduction of inflammation. So inflammation is a pathophysiological response to a number of triggers, usually infection, tissue injury, and so on. Inflammation actually, the name originates from the word flammo in Latin, which means flame. And the reason it's called that is that the 5 cardinal signs of inflammation, which were actually described more than 2,000 years ago, or nearly 2,000 years ago, by the Greek doctor and philosopher Celsus, who actually worked in Asia Minor, and are redness, heat, swelling, pain, and loss of function. A normal trigger for inflammation, for instance, that probably many people have experienced is some kind of injury to the skin or one of the barrier tissues in the body. I'm sure many people have been stung by a bee or got a splinter of wood in their skin, and as you experience that, you feel that the area starts swelling, turns red, and gets a little bit hot. And it's actually a normal response to the injury because your body wants to increase blood flow to the site of injury and deliver the white blood cells that actually protect us from infection and then help repair the injury to the affected area. And then the heat is also a sign of increased blood flow, but sometimes we develop fever, which tells the body there's something wrong. So actually there's nothing wrong with fever, because without fever, we want to know there's something wrong with us. Okay? So the good side of inflammation is that inflammation is very important for clearing infections, for regenerating the injured tissue, and the cells that manifest the inflammatory response, macrophages, neutrophils, and so on, in addition to killing bacteria, they come up to the injured tissue, mop up all the debris, and then secrete a variety of factors that stimulate tissue regeneration. So, in general, when you experience an inflammatory response, you should wait a few days and see if it goes away. But if it's really acute, it's something that you may have to go to the hospital for. But don't rush and just get some over-the-counter anti-inflammatory drugs, because a lot of time they may do more harm than benefit. So those of you who are interested in the regenerative aspects of inflammation, we published a review in Nature a few years ago, so I'm just bringing it up here, and I will not talk a whole lot about it. So the inflammatory response itself is very intricately regulated. It involves many different inducers. The inducers can be tissue injury, an infection, foreign body, which trigger a foreign body reaction. And then there are sensors and mediators. The sensors are white blood cells, such as macrophages, that carry and express receptors for the inducers of inflammation, and in turn, they release a variety of mediators of inflammation that can be inflammatory cytokines, such as TNF, IL-1, IL-6, but most rapidly, they release histamine, bradykinin, and a lot of small molecules that are responsible for the most immediate signs of inflammation. Now, most inflammatory responses are triggered within hours or sometimes even minutes of the injury, and they subside a day or two later. And if they last a few days longer, then there's some problem. But when the inflammatory response does not resolve, or the injury persists, or the infection persists, you can go from transient inflammation, acute inflammation, to chronic inflammation. And the problem with chronic inflammation is that it leads to activation of the adaptive immune system, T cells and B cells, and that can result in long-lasting inflammatory response, loss of tissue damage, and, for example, leads to initiation of diseases such as rheumatoid arthritis, inflammatory bowel disease, hepatitis, and so on, the kind of diseases that you don't want to experience. So I'll talk now about what I consider the bad side of inflammation. So by stimulating regenerative processes, persistent non-resolving inflammation can promote cancer, which is considered a disease of aberrant cell proliferation. However, it is estimated that at least 20% of all cancer deaths occur in the context of infection and inflammation. For instance, in China, there's a lot of viral hepatitis, or used to be a lot more, hepatitis B virus, which cause liver inflammation, but eventually can lead to liver cancer. Now we are preventing this with vaccines and access to more, safer water, safer food, and so on, but such diseases still persist. And I'll talk about some typical inflammation-driven cancers, colorectal cancer and liver cancer. But another very common cancer in China, actually the #1 cancer here, is lung cancer. And lung cancer can be caused by smoking, but also by exposure to air pollution. Cigarette smoke contains a lot of carcinogens that can cause mutations that eventually lead to cancer. But more than 10 years ago, we have shown that in addition to carcinogens, cigarette smoke elicits pulmonary inflammation, and the pulmonary inflammation actually facilitates the development of lung cancer in experimental animals. And more recently, work done by the group of Charles Swanton in Cambridge has shown that air pollution, which is a major cause of lung cancer, actually does not cause cancer by inducing mutations. It's actually causing cancer by irritating the lung and causing chronic inflammation. I think this is a very beautiful paper that they have published. So for me, if you look at different three blind scientists trying to describe cancer, which is the elephant, most of them think it's a genetic disease caused by mutations. In recent years, we have seen a trend to claim that cancer is a metabolic disease, but I'm somewhere down there trying to feel the trunk of the elephant, and I believe that inflammation also plays a very important in the development of cancer, and I will provide you with a few examples. So mutations, the genetic basis for cancer is very important, but many mutations are insufficient for inducing cancer. For instance, RAS oncogenes are some of the most common oncogenes. They are involved in colorectal cancer, pancreatic cancer, and many lung cancer, and so on. But we know, for instance, that pancreatic cancer, which is initiated by mutations in the KRAS gene, and it's one of the most difficult cancers to treat, it's usually deadly, is actually preceded by premalignant lesions that already harbor cells with KRAS mutations. And at a certain age, 70% of the population actually already have cells that are mutated and have KRAS mutations. It's not a reason for alarm. You should not lose sleep about it, because most of these lesions will never progress to cancer. And if they do start progressing, it takes about 30 years. So if you start manifesting these lesions when you're 50 years of age, maybe you'll get cancer when you're 80 or 90. However, most people will not develop cancer in response to such lesions. Another example is melanoma. Melanoma, a very common, very aggressive skin cancer, is initiated by mutations in the BRAF genes. But these mutations already occur in blue navy that are lesions on the skin that are actually latent, and they do not progress, and they stay dormant for many, many years. But in response to inflammatory stimuli, such as repeated exposure to sunlight, some of these lesions become inflamed and they start progressing. So inflammation is very important. Another evidence, before we even started our work on inflammation and cancer, is that was epidemiological studies that have shown that non-steroidal anti-inflammatory drugs, abbreviated as NSAIDs, including aspirin, one of the cheapest drugs that you can buy anywhere in the world, have a huge impact reducing the toll of breast cancer and colon cancer. So actually, aspirin has saved more people's lives than even some of the most sophisticated and the most expensive anti-cancer drugs. And when people started understanding how aspirin works, and it works by inhibiting cyclooxygenases, which are enzymes that generate inflammatory agents called prostaglandins, and people realize that the more important cyclooxygenase is COX-2 rather than COX-1. Some companies actually started developing COX-2 inhibitors to treat inflammation, and they're actually very effective. But then Merck carried out a clinical trial with a drug called Vioxx, which was very effective, reducing the risk of colorectal cancer. But turns out there were some other problems with this drug. And after several years of use, 1 in 10,000 people start developing cardiovascular events. But this was enough of a red flag for the US FDA to recall the drug, and now it's no longer marketed, and all the big pharmas got cold feet, and they will never attempt to develop preventive drugs for cancer. And in my mind, the most important thing in cancer therapy is actually to prevent cancer rather than to treat it when it's too late. So I think we need a change in the approach to cancer. So the question is, how does inflammation promote cancer? So we asked this question 20 years ago, or a little bit more, and we decided to study an inflammation-driven cancer called colitis-associated cancer. Colitis-associated cancer appears in people with ulcerative colitis or Crohn's disease, which are 2 of the most common inflammatory bowel diseases. It only accounts for about 5% or less of all cases of colorectal cancer. But as its name indicates, colitis-associated cancer is clearly driven by inflammation. So we decided to use mouse models of this disease and test the role of a transcription factor, which is a nuclear protein that regulates gene expression called NF-kappa B, in the development of this disease. So why did we suspect that NF-kappa B is important? Because NF-kappa B is one of the master regulators of inflammation. It is activated, it is sent from the cytosol to the nucleus in response to many different inflammatory triggers, as you see here. And once in the nucleus, it induces genes that code for a variety of enzymes that generate inflammatory mediators. I already mentioned COX-2. And it's also very important for triggering what we call innate immunity, which helps protect you from infections. It induces, for instance, a lot of antimicrobial peptides, chemokines, which are chemoattractions that recruit inflammatory cells to the site of inflammation. But at that time, we also found that NF-kappa B activation prevents programmed cell death or apoptosis. And it became clear that one of the most important defects in cancer is the resistance of cancer cells to programmed cell death. So programmed cell death is a way to get rid of bad cells. But cancer cells, which should be eliminated, have found ways to escape it, And we thought that NF-kappa B may be one of the important tools that cancer cells use to protect themselves from inflammation. So just targeting NF-kappa B alone was not all that useful because there are many different forms of NF-kappa B. So we worked for quite some time to actually understand how triggers such as TNF, IL-1, bacterial metabolites such as endotoxin or LPS lead to the activation of NF-kappa B. We identified a kinase complex called the I-kappa B kinase complex. What this enzyme complex does, it phosphorylates, puts phosphate groups on proteins that inhibit the activation of NF-kappa B, retain NF-kappa B in the cytosol. They induce the degradation of the inhibitors, and then the NF-kappa B dimers can translocate from the cytosol to the nucleus. So once we knew the composition of the kinase, and we knew that the beta subunit is the most important one, we generated mice in which we could ablate the gene, delete the gene for IKK beta in any cell that we wish to. And since we are dealing with colorectal cancer, we deleted the gene in the epithelial cells, the epithelial lining of the colon and the small intestine, which are the cells from which the cancer originate. But we also did an extra thing. Since we thought inflammation is important, we deleted it in inflammatory cells, in macrophages and neutrophils, that these are cells that do not accumulate any mutations. They are not genetically altered, but we postulated that they are very important participants in the cancerous process. And indeed, when we ablated IKK beta in the epithelial cells, we prevented the development of cancer because we prevented the expression of survival genes that protected the cancer cells from cell death. But also, when we ablated IKK beta in macrophages and neutrophils, we prevented the development of cancer. And we found that in different cells, NF-kappa B does different things. In the epithelial cells, it prevents cell death, but in the inflammatory cells, it's important for the generation of inflammatory cytokines that also act as survival factors that trigger pathways and processes that protect the initial cancer cells from cell death. And one of the most important survival factors turns out to be the inflammatory cytokine interleukin-6 or IL-6. And we have shown that if we now ablate the gene for the transcription factor, again, the regulatory protein that is stimulated by IL-6 called STAT3 in the epithelial cells, we also can prevent the development of cancer or get much fewer tumors, and you can see that the tumors are quite a bit smaller than the tumors that evolved in the wild-type mouse. And that principle of tumor-promoting cytokines playing a very important role in cancer has been shown again and again after our initial publication in many different forms of cancer. So it seems to be quite general. So after looking at colitis-associated cancer, we decided to look at the most common form of colorectal cancer, which is sporadic colorectal cancer, which is thought to be caused by mutations. But we found that what happens in sporadic colorectal cancer, which represents about 90% of colorectal cancers, that there is tumor-elicited inflammation. The development of the tumor, or the initial tumor, also called a polyp, and that's something that colonoscopies identified by colonoscopy results in perturbation of the intestinal epithelial barrier, which separates our tissues from the billions of bacteria that reside in our intestine. So the barrier is extremely important. It protects us from all the bacteria that are present in our body. These bacteria usually don't cause any problem unless you rupture your appendix. But when the tumor evolves, we found that it perturbs the integrity of the barrier, and now bacterial metabolites, especially endotoxin, can translocate across the barrier, stimulate tissue-resident macrophages, which then produce a variety of cytokines that play a very important role in the sporadic form of colorectal cancer. So inflammation plays an important role not only in inflammation-driven cancer, but also in the most common forms of colorectal cancer that a lot of people did not suspect inflammation to be involved. Now I'd like to switch gears and talk about another cancer, which used to be the second most common cancer in China, but now with development of vaccines and so on, we see that the incidence of liver cancer is declining. So we are working on the most common form of liver cancer known as hepatocellular carcinoma, or HCC. And in the US, we see that the incidence of viral-induced HCC is really going down rather quickly, but it's replaced by a metabolic form of liver cancer called NASH-associated liver cancer. NASH stands for non-alcoholic steatohepatitis, or metabolic dysfunction-associated hepatohepatitis. The name was recently changed. And what happens in this disease is you start accumulating fat in the liver. Currently in the US and Western Europe, about 30% of the adult population have fatty liver, and about 1/3 or 20% of those have actually steatohepatitis, which is hepatitis caused by fat accumulation or other metabolic defects. And that type of inflammation is as bad as viral hepatitis, and it increases the risk of liver cancer. So we've been working on liver cancer for quite some time, and nearly 20 years ago, we have shown that one of the most important processes in liver cancer, again using mouse models, is injury to the liver, injury to the hepatocytes, that kills the hepatocytes, and then they release a variety of triggers or alarmins or damage-associated molecular patterns that trigger regenerative processes in the liver. The liver is the most capable tissue of self-regeneration, but it's also the curse of the liver, because when you trigger regeneration in the liver all the time, you stimulate cell proliferation, and cell proliferation can lead to cancer. So we continue to work on liver cancer. We switched from carcinogen-induced models in mice to mice that more closely, models that most closely reflect MASH or NASH-associated HCC. And we have shown that inflammation and in response to a cytokine called TNF, tumor necrosis factor, plays a very important in the development of MASH. And more recently, we have shown that in addition to triggering the regeneration of hepatocytes, chronic inflammation in the liver has another very important effect. It leads to suppression of our anti-cancer immunity. So our adaptive immune system can actually recognize early cancer cells because they express different proteins, also known as neoantigens, and it can protect us from cancer. But chronic liver inflammation, instead of stimulating the adaptive immune system, can lead to suppression of the adaptive immune system, and we have shown that this can result in the generation of immunosuppressive plasma cells. These are antibody-producing cells, which are very common in different liver diseases, not only metabolic steatohepatitis, but also in viral hepatitis, alcoholic hepatitis. And these B cells secrete factors that suppress the activation of cytotoxic T cells, and the cytotoxic T cells are actually preventing liver cancer. And one way in which we could overcome the immunosuppressive effects of these plasma cells is by using checkpoint inhibitors or anti-PD-1 drugs. And we have shown that they are quite effective in the mouse models. And while we were publishing these results obtained in mice, the first clinical trial have shown that this type of drugs also work in human hepatocellular carcinoma. Unfortunately, the response rates are still a bit low. They're somewhere between 15 to 20%. We and other people are trying hard to find ways to increase its response rates. We described actually a drug that is used for treatment of high blood pressure called losartan, can actually augment the response to checkpoint inhibitors. But this is another story for another time, because I'd now like to switch to the ugly side of inflammation. So the worst side of inflammation was actually described by Paul Ehrlich, who is the father of modern immunology, working in Berlin and later in Frankfurt over about 100 years ago. And he described what is called the horror autotoxicus. So autotoxicus means that our body generates a toxic response to itself. And now we know that it's due to production or activation of adaptive immune cells. And one of the most common autoinflammatory diseases that mostly affects women is lupus. And lupus is a very bad disease. It can have some minor manifestation, but it can also lead to kidney damage, and it can be fatal. And despite a lot of research on this disease, we still don't understand the origin of the disease, and we don't really have any good treatments for the disease. We can treat the symptoms, but once you stop treatment, the disease flares up and comes back. So the only way now to treat such autoimmune diseases is actually to completely inactivate the adaptive immune system, which makes you susceptible to infections. But we need clever ways to actually figure out what's going on. So go back to chronic inflammation. And as I said, inflammation is normally initiated by the adaptive immune system in response to cell injury, by the innate immune system in response to injury or infection. But once it's chronic, the innate immune cells, macrophages and neutrophils, can lead to activation of adaptive immune cells, T cells and B cells. But exactly how, nobody knows. So a few years ago, we started working on the role of mitochondria, which are the cellular power plants. They provide our cells with a lot of ATP. ATP is essential for life. work by Xiaodong Wang, who's now in Beijing, have shown that mitochondria also play an instrumental role in cell death. They release cytochrome C, trigger apoptosis. But we looked at the role of mitochondria in inflammation, and we find that oxidized mitochondrial DNA, mitochondria are actually originating from bacterial parasites that populated some of the very first eukaryotic cells, and damage to the mitochondrial DNA can result in fragmentation of the DNA, oxidation of the DNA, and the oxidized mitochondrial DNA can leak out from the mitochondria, and then it wreaks havoc also within the cell and within the entire body, and that's what I would like to describe to you right now. So we've been working on oxidized mitochondrial DNA as a very important trigger of inflammation, but then we became aware that there are quite a few studies that find elevated levels of oxidized mitochondrial DNA in the circulation are associated with lupus. They are actually very good predictor of disease severity. When the disease is very aggressive, oxidized mitochondrial DNA goes up. When you administer therapy that reduces the severity of the symptoms, it goes down. And then when the disease flares up again, oxidized mitochondrial DNA goes up again. It's not only associated with lupus, it's associated with rheumatoid arthritis, and it's also associated with a variety of metabolic diseases, including NASH and type 2 diabetes. So, as I'll show you, it's not only a marker for the disease, it's a very important trigger of autoimmunity. So, the problem with lupus is somehow our body starts generating a lot of antibodies to self-components. The immune system is built to discriminate between self and non-self, and in lupus, there's increased production of antibodies to DNA, different nucleic acids, and components of chromatin. And exactly how these antibodies are generated is not clear. So this is still a mystery. So, as I said, oxidized mitochondrial DNA is generated in response to mitochondrial stress. Those of you who are interested, we describe the process, and it starts with movement of calcium into the mitochondria. This results in uncoupling of the oxidative phosphorylation chain, generation of free oxygen radicals, and then what they do is they can oxidize the mitochondrial DNA, but that actually requires stimulation of mitochondrial DNA replication, because normally the mitochondrial DNA is very tightly packed, protected by proteins, because our oxidative phosphorylation electron transfer chain generate reactive oxygen species all the time. And only in response to uncoupling, it goes up. So you have to protect the mitochondrial DNA, but when you stimulate mitochondrial DNA synthesis, it exists in a naked state, uncoated by proteins, and that's when it becomes susceptible to oxidation. And we have shown that there's a nuclease, an enzyme that cuts the DNA, that recognizes oxidized mitochondrial DNA, cuts it into small fragments, and these fragments can exit through channels in the mitochondrial membrane, reach the cytoplasm, and then they activate a protein machine called the NLRP3 inflammasome. So NLRP3 inflammasome is a rather complex structure. It has a core protein called ASC or ASC, and it has a sensor protein called NLRP3 that a lot of people were considering to be a receptor for different inflammatory stimuli, but turns out the stimuli that can lead to activation of the NLRP3 inflammasome are numerous. They include ATP itself, when it's released from the cell. They can include silica particles, urea microcrystals, cholesterol microcrystals, UV irradiation, and any kind of physical damage. And there's no protein on Earth that can actually respond to such different variety of stimuli. So we started with the hypothesis that maybe mitochondrial DNA or some other factors produced by mitochondria are directly responsible for NLRP3 inflammasome activation. I was lucky to have a very talented postdoctoral fellow in the lab, Zhenyu Zhang, who is now a faculty member at UT, University of Texas Southwestern. And what Zhenyu has shown, that there is a process called mitophagy in which our cells get rid of damaged mitochondria. And when he blocked this process, he found that NLRP3 inflammasome activation was prolonged. So this told us that somehow damaged mitochondria must release something that leads to activation of the NLRP3 inflammasome. And eventually, he identified oxidized mitochondrial DNA as the substance that directly binds NLRP3 and triggers assembly of the inflammasome and its activation, resulting in the production of the inflammatory cytokine interleukin-1 or IL-1, which was actually the first interleukin to be identified. And then another outstanding postdoctoral fellow is still in the lab, Hongshu Xian, also originally from China. She was trained in Singapore, though, and identified the entire process by which the DNA can actually be cleaved by a nuclease called FEN1, exit through pores in the mitochondria, reach the cytoplasm, bind to NLRP3, and activate NLRP3, and it's all been described. But then Hongshu came to me, and she said that one of the models that we use to induce the production of oxidized mitochondrial DNA in mice is based on injection of alum, which is a salt of aluminum, which is used as an adjuvant for vaccines. But when she injected mice with alum without any antigen into the peritoneum, she told me that there is swelling of the spleen. And the spleen got about 3 times larger after every single injection. So I told her, aha, maybe if you keep injecting alum into mice, you elicit chronic inflammation and you lead to activation of the adaptive immune system, because the spleen is the major site in which immune cells reside, both B and T cells. It's the major site for production of antibodies. And I told her, just keep doing that, and let's see if the mice develop an autoimmune disease. And she figured out the right protocol, and she has shown that repeated injections of alum increase the production of antibodies to DNA. Not only did it increase the production of the antibodies to DNA, they accumulated in the kidney, induce glomerulonephritis, which is actually the worst complication of lupus nephritis. And you can see we actually stained these antibodies to DNA and showed that they accumulate in the kidney, and not only the DNA, antibodies to DNA accumulate in the kidney, they lead to activation of a very important factor in blood clotting and so on called complement, and activated fragments of complement also accumulated in the kidney and giving rise to kidney damage. So then we had the model, and we decided to take a closer look at what happens. And before we saw kidney damage, we looked at the spleen in greater detail, and we found that the injection of alum induced structures within the spleen called germinal centers. Germinal centers are aggregates of B and T cells. They're organized in a very beautiful manner, and there are factories for the production of antibodies. And there's specific stains for germinal center B cells, and we could show that injection with the oxidized mitochondrial DNA induces the accumulation of germinal center B cells, the cells that stain in blue, and this paralleled the increase in the titers of antibodies to DNA. So then we asked, how does this happen? And without getting to all the gory details of the process, we identified that there's a very specialized population of innate immune cells called dendritic cells. These are specific dendritic cells called plasmacytoid dendritic cells. And what they do is they pick up the oxidized mitochondrial DNA in a form bound to immunoglobulin. That we always have some immunoglobulin in our serum. There are receptors for immunoglobulin called Fc gamma receptors. They internalize the DNA, deliver it to a structure called the endosome. In the endosome, it's recognized by another receptor called toll-like receptor 4, which is a very important component of innate immunity. But the activation of TLR9 leads to activation of NF-kappa B, activation of the NLRP3 inflammasome, production of IL-1 beta, which we identified as a bridge between innate immunity and adaptive immunity, and what we think it plays a critical role in the switch from chronic inflammation to autoimmune diseases. And hopefully we can initiate some clinical trials with available drugs to IL-1 beta and some other components of this process, and see if this can treat lupus patients, maybe develop the progression of the disease, but this remains to be done. So with this, I'd like to close and give credit to the group of people who actually was engaged in the last part of my talk, the work on autoimmunity. This is Zhenyu Zhang, who actually started the work on oxidized mitochondrial DNA in the NLRP3 inflammasome in my lab. This is Elsa. So Zhenyu moved to UT Southwestern. Elsa Sanchez-Lopez, a postdoctoral fellow from Spain, stayed at UCSD. She now has her own faculty position in surgical oncology. And the two postdoctoral fellows who did all the recent work was Hongshu Xian that I mentioned, and Kosuke Watari, who helped her with a lot of the flow cytometry that was necessary for the work. And thank you for your attention. I'll be happy to answer any question that you may have.
麦可卡琳院士的精彩演讲，现在进入问答环节。有疑问的同学可以举手进行提问，欢迎大家踊跃提问。
Thanks to Professor Michael KARIN for his wonderful speech. Now we will have a Q&A session. Students can raise up your hands to ask some questions.
Thank you, Professor Michael, for presenting us this interesting lecture. So I have a question to ask is, in the context of the link between inflammation and cancer, are there any preventative strategies individual can adopt and to lead to a healthier, cancer-free life? Oh, so So in the context of the link between inflammation and cancer, are there any preventative strategies individual can adopt to lead a healthier, cancer-free life?
That's an idea. So, as I said, it's currently estimated that about 20% of all cancer cases, or actually cancer-related deaths, the number of cancer cases is much larger. Our inflammation plays a very important role. So, if we could prevent colitis, viral and metabolic hepatitis, inflammation to the lung caused by smoking and air pollution, only by behavioral modification, accommodating a better lifestyle, eating a better diet, and so on, we can probably reduce the toll of cancer by about 20%. But that seems simple. Turns out that behavioral modification is the most difficult thing to achieve. People rather take a pill than change their diet or start exercising. Fortunately, in the case of viral hepatitis, we have vaccines. We have some treatments for HCV, for which there are no vaccines. But a lot of work remains to actually prevent cancer by attacking the inflammatory component in the cancer. So hopefully, people like you, the new generation of doctors and investigators that you are training here, will actually take this charge and lead us to a better life, cancer-free life.
Thank you. Are there any students who have questions?
Question over there.
So thanks so much for your fruitful talk. And I have a question about the HCC and the linkage of the colorectal cancer after hearing your talk. In Professor Francis Chan from the Chinese University of Hong Kong, that he have mentioned the M3 bacterium, which is a group of bacterium that can initiate or be a biomarker for the colorectal cancer. So I'm thinking that in the HCC, is it possible to, is it altering the composition of the biosalt? So the good bacterium in the gut is decreased and also increases the bad microbiota inside the colorectal that would initiate or further initiate the colorectal cancer? Both. Is it possible? Is it any relationship between them because of this complicated biosalt mechanism?
So there's a lot of work on what's called the microbiome or the microbiota, which are the billions of bacteria that live in our intestine and colorectal cancer. One problem with these studies is that different groups identify different bacteria as being responsible for colorectal cancer. But the normal way in which these experiments have been conducted was to either generate germ-free mice without any bacteria, or treat them with antibiotic cocktail that gets rid of most of the bacteria, and then monocolonize them with a single species, okay? And different investigators decided to pick different species that they suspected. But by doing that, you increase the concentration of this bacterial species by 10,000-fold, 100,000-fold, and I don't think this is very physiological. And it's the same way of giving a suspected carcinogen that is present in extremely low levels in the environment, and people found that some thing that is sprayed on apples contain a potential carcinogen. But when you take that substance and increase it 100,000-fold, yes, it can cause cancer. But does that mean that it causes cancer in real life? I don't know. So the proof would be that if you can develop a vaccine against these bacteria and prevent the development of colorectal cancer, we find that yes, liver cancer can also depend on bacterial products because the liver has a specific type of circulation called the portal circulation. The portal circulation is very important for the absorption of macronutrients from the intestine, delivering it to the liver, which is the major site of metabolism, but also endotoxin from the intestine is drained to the liver, and another important function of the liver is to get rid of the endotoxin. But I think the endotoxin is important, but whether individual bacteria are as important as the endotoxin they produce remains to be seen. So I don't know if that answered your question, but I thought that's related to what you asked.
Thank you, Professor. So the last question. Oh, okay. The teacher.
Yes, Professor Michael KARIN, very exciting talk. So I have two questions for you. So based on your slide, I find that in the later 2 or 3 slides, you're talking about the mitochondrial DNA release as well as the oxidation. So in the meanwhile, does that mean in your current research interest is also related to the mitochondrial DNA, related to inflammation? This is the first question. The second question is, so because I'm also working, I'm your follower, also working on immunity. So recently we found that the aging person, including the elderly, as well as the rheumatoid arthritis patient, they have the high accumulation of the DNA fragment. So we also generated genetic animal to demonstrate it's related to the inflammation. So what do you think about if we can control inflammation? Is this the way to anti-aging as well? Thank you.
Okay. A very important question. I wish we could prove that this is the case because Lenny Guarente at MIT, one of the pioneering of aging research, actually postulated and provided evidence, but actually from yeast studies, that mitochondria are very important for aging. And mitochondrial defects may drive aging. And mitochondrial defects may result in the generation of oxidized mitochondrial DNA. So I think we need epidemiological studies to measure the concentration of oxidized mitochondrial DNA in the general population and see if it increases as people age, and if some of the chronic inflammation, actually low-grade inflammation that you see in older individuals, maybe part of it is due to the accumulation of autoantibodies, including antibodies to DNA. By the way, the oxidized DNA, mitochondrial DNA, is not an antigen. It works by activating the plasmacytoid dendritic cells, so it activates innate immune cells, and then it leads to increase in the production of antibodies to DNA. So I think the antibodies to DNA, which are still a mystery, I think they're actually part of our ancient repertoire of antibodies that are produced all the time. They may protect us from bacterial infections. So I think they have some kind of a function, but they need to be produced in very low amounts. When you produce too much, these proteins can simply get stuck in the kidney and destroy the kidney. So what you ask, it seems very attractive, but we work mostly with animal models, but we'll be happy to collaborate with any clinical investigator who is actually interested in addressing this question.
Thank you. Thank you, Professor. We are very grateful to Professor Michael KARIN for answering the questions. At the same time, thanks for Professor Michael KARIN again for coming to our university to give us a wonderful speech in spite of the busy schedules.
有请公关同学为麦克卡琳院士赠送鲜花。
Thank you, Professor Michael KARIN. Please be seated. Thank you so much.